Grd10s, how's it going? Alright, so we're going to continue off of the nucleus. So I just want to remind you about what we did in the last lesson. We spoke about the nucleus and how it's the brain of the cell. We also discovered that it has quite a couple of important parts. And if you really are thinking about the fact that you don't need to study it, your entire first section in matric is based around the nucleus as well, and all the genes and DNA and so on and so forth inside of it as well. So please make sure that you fully understand it and that you draw it, and then you can draw it off by heart because it will be in your cycle test. We also did a whole introduction to organelles again, and well, that was in the first one. And I just want you to recognize that we're just continuing with it. Remember, organelles are the functional parts of a cell, and that help the cell actually function and survive as a whole. All right, so now, today we're going to start with the cytoplasm. So this is the part of the cell that is enclosed within the cell membrane. So I put a little picture down below over there, something you guys would probably be able to draw. I don't draw this. I'll get you to draw it later. So it is enclosed by the cell membrane, and that just means it's this liquid which holds everything inside. So it says that it contains all the organelles, which means it holds it or suspends it. So you can also know the cytoplasm as a suspension. Remember I said it's a bit more of a jelly-like substance. Then the contents of the cell nucleus are not part of the cytoplasm. And we mentioned earlier that there's the nuclear plasm around the nuclear membrane around the nucleus, so that there's no connection between what else is in the cell as well as what's in the cytoplasm, so that it can't be affected. So it's the site where most cellular activity occurs. And please remember that the cytoplasm is majority made out of water. It's probably around 75% water, which means that, and if you remember from water when we did inorganic molecules, you'll recognize that's where I actually said that majority of the chemical reactions take place. So majority of the chemical reactions take place. Chemical reactions are cellular activity, and if the cellular activity occurs in the cytoplasm, then boom, the cytoplasm is made out of water. All right, so I put two examples in there for you: glycolysis from cellular respiration and cell division. Cell division is your next section of work. All right, so the cytoplasm, not too crazy. You can see that in the diagram I put over there, it's the light blue mixture that is all around the organelles, and then the black dot, and the thing around it would be the nucleus and the nucleolus, just in case you're wondering. All right, now I need you to put your brains on, please. The cell membrane is probably as important as the nucleus is itself, and it is very important for grade 10, and then I expect you to be able to use the cell membrane quite freely throughout the other sections of work that we're going to do. All right, so now it's otherwise known as the plasma membrane, and we can also call it the cell membrane, plasma membrane, and very weird scientists call it the fluid mosaic model. So if any of you do mosaic, you'll know that it's on the sides of your pool where all those little tiles are, and they generally make a shape of a dolphin, or people use it to make man, sorry, my nose is itchy. Make a whole bunch of things really pretty. It's just tiles that are broken up and then we use it put together. So that's according to what this is. So the fluid mosaic model is made up of phospholipids, proteins, and carbohydrates. So I put a very complicated version of it in there just so that you guys have something to reference to as we go through the cell membrane. Because what you need to recognize is that the word fluid is inside over there. So please remember that this is made out of lipids. Remember the phospholipid. So you can see in the bottom right hand corner over there, it actually has a labeled phospholipid over there. Then there's a whole bunch of things. There's the blue things, there's the orange things, there's the yellow things, and there's the green things. I'll get to what each one of those are as we move along, but what I really also want to point out is that a lot of the time when I'm going to talk about the cell membrane, I'm going to talk about the extracellular fluid and the intracellular fluid. So intracellular fluid is otherwise known as cytoplasm, and you'll see right at the bottom of the slide there's a little red label saying cytoplasm. And that you'll be able to pick up is the inside, or what's on the inside of that cell. So the blue stuff from the previous slide. And then extracellular fluid is any fluid that is on the outside of the cell. So extra not as in it's being crazy, extra as in outside, and intra as in inside. Okay. There you'll see stuff you can see carbohydrates at the top right hand corner of that diagram. You'll see hydrophilic, hydrophobic. You'll see some surface proteins. integral proteins, cholesterol, as well as protein channel. So now, remember when I said to you that cholesterol is actually part of the cell membrane as well, and that forms part of the fluid mosaic model. What I want to point out here is that cholesterol actually causes your cell membranes to be more liquid-like, and fewer or less cholesterol causes it to be more solid, if I can put it that way. It's just, it's like more rigid. It doesn't move as much, you know. And I want you to think about, look at that huge integral protein right in the middle over there. So now imagine I was on a cold day, and we know that when things are colder, they shrink, and when they're warmer, they expand. So do you think you can put more proteins in when the cell membranes themselves are really hot, because there's big spaces in between them? So now if it becomes really cold, all of a sudden everything squeezes, and the phospholipids aren't going to move, but the protein itself is going to pop out. Now that leaves a huge gaping hole in your cell membrane. So always just remember that this is quite fluid, and it does stop the movement of fluids in and out. So please just, like, think about this thing as something that is changing and moving constantly, and that's ultimately why it's called a model. Okay, so this is the fluid mosaic model. This is a very, 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 very basic version of it, just to help you from the previous slide. So it says there the hydrophilic region, it's water-loving, and the hydrophobic region, which is water-fearing, and then there's a transport protein in over there. You'll also notice that beautiful little green thing floating in the back over there, and I want you to put that in your mind already. That is like an aerial. It almost looks like a whole bunch of kites being flown. And those kites are actually a way of the cell to communicate with whatever's on outside of it. Because you can imagine that the cells need to communicate with each other, tell each other what's going right, what's going wrong. Maybe it's become hot, maybe it's become cold, maybe it's become too acidic, maybe it's become too basic. And those little receptors are actually able to tell everything what's going on, and they are called a glycoprotein. So you can see it's this mixture of a whole bunch of different bits and pieces that actually make up the fluid mosaic model, or the cell membrane, or the plasma membrane. So I need you to stop the video here, and I want you to draw this. It's extremely important. Please don't worry about where the labels go. I will never expect you to draw this entire thing. What I will expect you to do is draw a phospholipid, which I have later on in the slides. So don't worry too much about that. But I mean, long story short, if I did ask you to do a diagram, it would just be that sort of browny red color. And then the yellow streaks at the bottom of that, which we drew in the previous section anyway. So what is important over here is you can see the glycoprotein, the glycolipid on the top right hand corner. There's a protein molecule, hydrophobic tail, internal surface membrane. I don't need you to know that. But I do expect you to remember the hydrophobic tail, so please do not write internal surface membrane. Then there is a carbohydrate chain, which is used for the communication between the different cells. And to a large extent, they can even use those carbohydrate chains to actually hold on to each other. So, because remember, you're not just melting apart. There has to be some way that all your cells are kind of holding on. There are other ways of doing it, but we'll get to that later. So then you have the external surface membrane. Please don't worry about that. Then we have the hydrophilic head, and then there's a new word I need you to learn. It's called the phospholipid bilayer. I have a better diagram in the next slides, but please just note it's like bi meaning two layers of phospholipids. And how are those tails arranged? The tails are facing towards each other, where the heads are facing away from each other. So the water is attracted to the outer surfaces, but it is not attracted to the inside, because that is hydrophobic. So as the water moves into one, it actually bounces around on the inside and then gets attracted to the other side and spat out. And that's what allows this whole thing to be semi-permeable as well. So please remember that when I mentioned semi-permeable, I said that it has to be directly through a membrane. So after you've drawn that, the cell membrane, each phospholipid has a swollen head and two tails. The head is hydrophilic and the tail is hydrophobic, and it's arranged in a lipid bilayer. So now you've got a couple of words all meaning exactly the same thing. You've got cell membrane, plasma membrane, fluid mosaic model. Same. You now have phospholipid bilayer and the lipid bilayer. Both of those exactly will mean exactly the same thing. You just need to remember the whole idea of what a phospholipid is. It is a lipid, and remember that a lipid has 3 fatty acids and 1 glycerol. And I'll show you in the next slide about that now. But so it's made out of lipids, and it's a bilayer. There are 2 layers of it. Does this mean that it is double membraned? No. It just means that it has 2 layers. If you had a double membrane, that means you would have 2 phospholipids, like you would have 1 phospholipid there and another phospholipid bilayer over there. So 1 bilayer, 2 bilayer. And then those going around like that would be the double membrane. This is just a single membrane made out of 2 layers. Alrighty. The heads are pointing outwards, either extracellular or intracellular. Remember, extracellular facing the outside of the cell, intracellular meaning cytoplasm or facing the inside of the cell. This is for you little smarty pants who refuse to just take my word for it. You do not need to learn that entire top structure over there, but I hope that you recognize parts of it. So you can see that those little bits and pieces on the top over there, there are 2 chains, 2 carbon chains. These are the fatty acids. So the top fatty acid is completely saturated. It has no double bonds in whatsoever, whereas the second fatty acid is unsaturated. Okay, and that's why it has that kink in it, and you can see right where that bend occurs, there is a double bond directly over there. Then where the third fatty acid should be is now on the left hand side, and there you get to see that beautiful little phosphate group over there, which is in that slightly darker blue. And then just remember that that phosphate group, hey, sounds a lot like ATP, so adenosine triphosphate. So the way that it actually attaches is by the use of energy as well. Then this is the common drawing that we always have of it. You can see the polar head and non-polar tails. Please don't worry about polar and non-polar. You just need to know that the polar head attracts water, always hydrophilic, and the non-polar tails are hydrophobic. And then when you put one tail to another tail with the heads on the outside, that creates the cell membrane, and that as a whole is called the phospholipid bilayer. Alrighty, so now we have the cell membrane again. So we have protein molecules spread between the phospholipids. So in the diagram below, I do not expect you to draw it either. I want you to recognize that there are these big orange structures all over the place, and you saw it right in the beginning of the slide set as well, that there are these proteins that are spread around. These proteins are extremely useful. If you did not have these proteins, you wouldn't be able to function. Your cells would not be able to communicate. You would not be able to accept certain molecules. Your nerves wouldn't work. There's a whole list of different things if you did not have those exact proteins. And that is in between the phospholipids. And you can notice as well that some of the proteins go directly through the cell membrane and some of them sit on the surface. So for instance, the bottom left hand corner, there's a peripheral protein, and peripheral means on the surface, whereas you have integral proteins, which is a protein that goes all the way through the entire structure itself. Okay. Then you're going to notice those little green things on top of all the orange things. So those are carbohydrates often attached to the external surface of certain proteins, and they may join cells together, which I mentioned earlier, and as well act as sites where hormones or chemical messengers can attach. So for those of you who are already thinking like, okay, well, how does this affect me? Anyone who has diabetes, their glycoproteins do not function. So you're either born without the genes to create the glycoproteins, or it's just the idea that your body can't read them. So this leads to diabetes, where the insulin can't force the cell to take any of that glucose in. And if it can't force the glucose to come in, then, well, your body doesn't know what to do with it, and then eventually it just spits it out, and then you end up with diabetes. All right, so let's just look at the functions of a cell membrane. First, it is selectively permeable. So proteins form pores or channels to allow certain substances through. So I have put a picture in there of a transport protein. And remember, in active transport, we went into transport proteins in a bit of detail, where I explained to you it was the use of ATP that forced molecules to move through. Some of these proteins bind to substances to carry them to the other side. So let's just quickly go over the definition of active transport. It's the movement of solutes against a concentration gradient with the use of a transport protein and ATP. Boom. All right. Then next, it controls the flow of information. Previously, I spoke about how we have those glycoproteins, those red things in the previous slide, and they are known as receptors. Now, what is a receptor? A receptor is anything that is able to receive a message. So receptors are really important. So receptors have the ability to read and generate signals. Therefore, it plays a central role in the biological communication. And it's not just the communication between cells, it's also the communication of whatever you've put in your body that now needs to affect the cells as well. So don't just think like these things are very specific in like how or what they speak to. It's the idea that there's like a whole idea of communication as a whole. Then we have the functions of the cell membrane, and you'll be able to see that beautiful little cute pink thing busy eating cake. Okay, an animal cell membrane is able to take up liquids in pinocytosis and engulf solid food particles by phagocytosis. Some people say phagocytosis. It's completely up to you. The word phago means to eat, cyto meaning, or cytosis means of the cell. So this is eating of the cell, and you can see that this little cell over here, the cell membrane actually like, if the cell was like that, it actually starts to like open and grow around what it needs, and it actually can bring it inside, and then it can digest it. Okay, so anything more than that, I'm 100% with. I am proud of you ladies for getting through all of this, and let me know if I can help you anymore. This will be the end of this lesson, and then next lesson we'll be doing a chloroplast and mitochondria. There's a lot of work in there, so the lesson's going to be a little bit longer. But I hope you're doing well, Grd10s, and I'll chat to you soon.